right, so remember that sexual reproduction involves first thinking about meiosis, so the production of haploid cells, eggs in the female, and the production of haploid cells, sperm, separately in the male, and then thinking about the two coming together in fertilization. So I give you a little practice thinking problem. It says the diploid number of a lion is 38. How many chromosomes are in a lion sperm? And how many chromosomes are in a lion egg? So if the diploid number of the species is 38, they're telling you how many chromosomes are in a typical diploid cell, typical body cell. And we know that sperm and egg are gametes, the products of meiosis, so those have half the normal number. So if the diploid number of the lion of two sets of chromosomes is 38 chromosomes, then one set of chromosomes would be 19. So a sperm has 19 chromosomes, an egg has 19 chromosomes, and when a sperm and an egg fuse together, both of those nuclei fuse together as well, and you get a new nucleus that has 38 chromosomes again. So in each generation, the offspring have the same number of chromosomes as the parents. Now, the next question. The diploid number of the tiger is 48. How many chromosomes are in a tiger sperm? In that case, one set of chromosomes would be 24. So a tiger sperm has 24 different chromosomes in it. A tiger egg would have 24 chromosomes in it. How many chromosomes would be in a tiger zygote? Well, if a sperm with 24 fuses with an egg with 24, then the new zygote has 48, just like the parents. All right, now here's where we get to something interesting that really requires you to think through this in steps. If a male lion and a female tiger mate, how many chromosomes are in the zygote, which is called a liger? I'm not 100% sure on the spelling there, but all right, so we need to think about meiosis first. A male lion makes sperm. How many chromosomes will be in the sperm? Well, we said the diploid number of the lion is 38, so the sperm would have 19. A female tiger, the tiger diploid number is 48, so the egg would have 24. How many chromosomes are in the zygote called a liger? That would be 19 plus 24, so it would be 43, which is weird, but that is the answer. So the implications of that we can talk about in a later chapter, but I'm just asking you to go through this thought process about how many chromosomes would have to be in the egg, how many chromosomes would have to be in the sperm, and then fusion of those, you just add them together. So go through the same thought process for a cross involving a horse that has a diploid number of 64 and a donkey that has a diploid number of 62. If the diploid cells go through meiosis to make gametes, you would have, it's a male horse and a female donkey, I think. If you had the female horse and the male donkey, it's called something else, hinny, I think, but I might be switching that around. But in any case, if you had a cross between a horse and a donkey, how many chromosomes would be in the horse gamete? How many chromosomes would be in the donkey gamete? How many chromosomes would be in a mule? Well, the horse gamete would have 32. The donkey gamete would have 31. So the mule would be those two added together, 63. Weird, but true. So I will ask you some questions like that to make sure that you're thinking about meiosis first, separately for the production of the egg and the production of the sperm, and then add those together to kind of represent the fusion process, fertilization. All right, the rest of the chapter is a little bit of terminology and concepts. One of the main advantages of sexual reproduction is that it produces a lot of genetic variation within a species. And the number one source of all genetic variation is mutation, where DNA is mutated, meaning little changes in the DNA that are caused sometimes by the DNA replication process. Mistakes during DNA replication can create a lot of mutations, but also things like UV and X-rays and chemicals that come into your body from the outside world environment. But no matter where the mutations or how the mutations are caused, mutation creates different versions of genes that we call alleles. And the alleles of the different genes are recombined into many combinations by the process of crossing over, which happens in prophase 1 of meiosis, and then something called independent assortment, which is primarily associated with metaphase 1. So briefly, we're going to look at crossing over. Crossing over creates new combinations of alleles, specifically for genes that are located on the same chromosome type. So in this image, they're just focusing on the long chromosome, a homologous pair, one they colored red and one they colored blue, but that's a homologous pair of chromosomes. And let's say you have gene A at one end of the chromosome and gene B at the opposite end. And let's say you had two different alleles of gene A in this cell and two different alleles of gene B in this cell. Now, we said that when meiosis starts, prior to prophase 1, you have a DNA synthesis similar to S phase that we saw in the cell cycle. So there's a DNA synthesis, a replication that makes the chromosomes dyads. And then one sister, I guess you could call it, one sister here and one sister here. So we call these two non-sisters. These two here are non-sister chromatids, but they're on homologous chromosomes. They break and rejoin, and you can see that they're trying to show that right here. There's a breaking and exchange of equal corresponding segments, and then a rejoining, a connection back to one of the two or both of the chromatids. And so in the end, after meiosis 1 is finished and then meiosis 2 is finished, over here you see the overall results. One of the final gametes doesn't have anything kind of unusual about it, and this one doesn't have anything unusual about it. We call those parental allele combinations. In other words, one of the homologous chromosomes in this parent already had a big A and a big B on the same chromosome, and this gamete has that same thing. One of the chromosomes in the parent cell, in the original cell, I should say, had a little a and a little b, and we see that in this gamete. So these gametes aren't so interesting. It's these middle ones that have the new combination. In the original cell, there was no single chromosome that had a little a allele connected to a big B, and there was no whole chromosome originally that had a big A connected to a little b, but now in the gametes there is. So that's crossing over creates some new combinations of alleles for genes that are located on the same homologous, same chromosome itself. Then we kind of take that concept and we overlay it with this concept. Independent assortment primarily describes the way that the homologous pairs line up in metaphase 1 with respect to each other. So this is the long pair of chromosomes, this is the medium pair, and this is the short pair. At least that's how I'm going to call them. So the long pair of chromosomes, when meiosis happens, it could be that the red one, and remember, these don't actually have color, but the red one could be on the left and the blue one could be on the right. And that, by coincidence, could be true of the medium pair, and it could also be true of the small pair. However, doesn't have to be that way. When meiosis happens in another instance for the same organism, you have a possibility of the red large chromosome on the left and the blue large one on the right, but maybe down here for the small chromosomes, they're switched, and that's perfectly fine. Or maybe these two are both switched compared to this. And so you have all these different ways that the homologous pairs can line up, independent of each other. And so ultimately, the gametes that you get have all the different combinations possible. And we want to think of these blue and red, sometimes called maternal and paternal. All of these chromosomes belong to the same individual. But in a way, half of them came from the egg, so half of them belong to their mother, and half of them also belong to their father. All the chromosomes are theirs, but at the same time, they're half maternal and half paternal, referring to their parents. So what they pass on to their children are some chromosomes strictly just from their mother, some chromosomes strictly from their father, some combinations of that in various forms. The only rule for meiosis is that each gamete has to have a full set, and a full set in this example is one large, one medium, one small. And so independent assortment creates lots and lots of possible combinations for that haploid set of chromosomes that's in the gamete. So in humans, what this means is that because we have 23 pairs of chromosomes, independent assortment alone produces 8.4 million possible chromosome combinations with regard to maternal and paternal combinations in any one gamete. 8.4 million possible combinations. Then two gametes have to fuse to do a fertilization event, and any two gametes fusing together would have a probability of 8.4 squared, 8.4 million squared, and that produces 70 trillion combinations of diploid zygotes. And then one thing we really can't quantitate is the crossing over. Crossing over, if we consider the crossing over, which happens at least once for every homologous pair of chromosomes in the cell, and it occurs randomly every time meiosis happens, what we do know is that the true amount of genetic variation among humans becomes astronomically large. And that means that no one else has the exact same combination of all the alleles that you have, with the exception of those who have an identical twin, because identical twins come from one zygote. They're called monozygotic. And this fact is going to be important when we talk about genetic testing for identification purposes, sometimes called DNA fingerprinting, for forensic testing and paternity testing. All right, so wrapping up, comparing mitosis and meiosis, make sure that you are very clear on the purpose of mitosis and the purpose of meiosis, and the products of mitosis versus the products of meiosis. So what is the purpose and what are the products? I've gone over that in the lecture. And also, there are several events in meiosis that do not occur in mitosis. All of them occur in meiosis 1, and these events I will mention briefly. In prophase 1, you have synapsis and crossing over. In metaphase 1, you have the lining up of the homologous pairs of chromosomes at the metaphase plate. And because of those things, you have a huge amount of genetic variation in the gametes. And so those are two critical events that occur in meiosis 1, and they don't occur in meiosis 2, and they don't occur in the cell cycle mitosis. So be very clear on those special events.